Good afternoon, everybody. It's so great to see so many names on the screen. I'm Ellen Hahn, the director of the UK Center for Appalachian Research and Environmental Sciences. I see many faces that are names I recognize. I'm going to turn this over to Kevin in a minute. I just want to give you a commercial break about visiting our website and using our portal if you need services or need consultation on your research. We are just waiting to help you. So I want to introduce Kevin Pearson. Many of you know Kevin. He probably doesn't need any introduction at all, but Kevin does lead our career development program at UK Cares, and he will introduce our esteemed speaker, Dr. Peter Nathanielsz, and we're so excited. Thank you.
Yeah, so thanks, Ellen, and I want to thank Dr. Peter Nathanielsz for joining us today. He is joining us from England, so he's probably our farthest Zoom so far. We've had some from all across the United States, but he's currently in Europe, so that's even further. So it's been my pleasure to invite Peter to come talk to us today, and he's going to introduce a topic that is near and dear to my heart, which is developmental programming, and he's a world's expert on this field. He's written a very popular book, and now a second edition of his book called Life Before Birth, The Challenges of Fetal Development. So Dr. Nathanielsz, he actually earned his bachelor's, PhD, MD, and Doctor of Science from St. Catherine's College in Cambridge. He then started as a faculty member at the University of Cambridge and then has gone to many prestigious universities within the United States, including UCLA, Cornell, New York University School of Medicine. And Peter does a lot of work with non-human primates and does developmental programming work in that model. So then the whole unit moved from New York to Texas in San Antonio at the Texas Health Science Center. And then his whole group then again moved to the University of Wyoming, where he's currently adjunct faculty there. And Peter has published over 600 manuscripts, and I actually met Peter about 10 years ago, but I knew him for much longer. So I don't want to take too much of his time, but I just want to give you a little bit of how he's really a mentor and a really hero, a scientific hero of mine, which is that Peter and I met at a meeting in 2011 in Portland, Oregon. And I went up to him because I did a postdoc in something that was very different in the research that I do now. So all of my scientific heroes in developmental programming, I just learned from reading on PubMed, and Peter has published over 600 papers. So I read a lot of his papers because he's an expert fetal physiologist. And it just so happened that a month or so later, he came to Alltech and gave a talk. They used to do a really nice nutrigenomics seminar in Lexington. And I met Peter and actually really interacted with him, engaged with him at that meeting. And we really started to foster a relationship. We've never published together, but we've enjoyed communicating about our research. Peter's been funded since 1976 continuously from the NIH. He currently has U19 from the National Institute on Aging, which is a huge grant. It's almost $3 million per year to keep his non-human primate colony going. And I was actually fortunate enough to see Peter receive the pretty much a President's Achievement Award at the Scientific Conference for Society of Gynecological Investigation, which is now Society for Reproductive Investigation. And that was in Orlando, Florida. And it felt great to me to be at a conference with pretty much the keynote speaker for the entire meeting. And he and I were able to have lunch together and enjoyed further communications. He's won numerous awards, been a Fulbright scholar. And I'll just tell you this one funny story, and then I'll let Peter take over. But I was a new assistant professor, right, hanging out with one of my scientific heroes. And I just thought the world of him. And I finally published my first paper where I was corresponding author, looking at maternal exercise and animal models. And literally a week after that paper comes out, so this is 2012, about a year after I met Peter, I get a voicemail on my office phone, which never rings. And it's a voicemail from Peter, and it's like, "Hey, Kevin, I really need to talk to you." And I'm like, "Oh my God, he saw my paper. He hated it. Like, he didn't like it, whatever." So I'm like, I get that heart racing feeling, and I give him a call, and he's like, "Hey, I was having a conversation with my friends, and they didn't believe that you could get good scotch out of Lexington." He's like, "Can you go down to Alltech and get the Pierce Lions Special Reserve and bring me a bottle the next time you see me?" And I'm like, "Absolutely." And my heart finally came back into my chest. So Peter, I hope you and your friends enjoyed the scotch. And I cannot wait to hear your talk about this book, which I have actually purchased. And I now, it's going to be required reading for all students that come into my laboratory. I've told them that I will pay for any of the books that they buy, because it's such a great summary in layman's terms about pregnancy and developmental programming and how we can really foster a great environment in the in utero period, which will lead to hopefully a lifetime of health versus a lifetime of disease. So Peter, please take it away.
Well, thank you very much, Kevin. That's extremely generous of you. Since you're recounting anecdotes, my great hero, who I did sabbatic with twice, was Mont Liggins, the New Zealander who did the basic research on surfactant and whose work has in fact saved thousands of premature babies' lives. And I was once at a meeting when he was introduced in similarly extravagant terms, in his case merited, in mine not so much. And I thought, what on earth is he going to say? And he just said, well, he said, of all the introductions I've ever had, that was the most recent.
That's funny.
Sorry about that. Perhaps it didn't come over as a joke.
Oh, we ask everyone to mute during the meeting, so you'll only hear me. They turn off because just to keep the broadband or the message going.
Sure. Okay, well, look, what I am trying to do today is to tell those of you who know nothing about fetal development and developmental programming something, and then perhaps also to tell some of you, to discuss with some of you things who already know in many instances more than I do. So let's sort of get going. Oops, now of course. I thought I'd put a reason or two why I wrote the book, because for me, there's nothing more fascinating in the whole of biology. How we pass from one cell to the first million trillion cells. We pass more biological milestones before we are born than we will pass in the rest of our lives. The fetus is living by one set of rules and preparing to live by a completely different set of rules. It is a remarkable feat of physiology. Fetal physiology is just so exciting that I certainly am very pleased that I chose it. So developmental programming first sort of came to light in the 1990s by a guy called David Barker in the south of England. And mostly it was a human epidemiological story. There's just not time to go into everything. I strongly recommend you go into that story. But what it led to was the wish by many investigators to find real hard evidence, because human data, as those of you who work in human studies know, is beset by confounds, and we can get much more in many ways from the animals. So developmental programming is the fetal response to a specific challenge during a critical developmental time window in its fetal life that alters organ development with resulting effects on offspring's health that can persist throughout life. And there are, of course, many, many challenges that one can think of. And perhaps the most commonly investigated, because mostly they are the most common that affect pregnant women and families, are the challenges of maternal undernutrition, overnutrition, stress, and now I've put in environmental pollution at Kevin's request, though I'll tell you right off the bat, I don't know too much about it. But one of the useful things of us all getting together so often is that I've certainly determined that I'm going to try and do some more on developmental pollution. And I have included a few items. So if we look at maternal obesity to start with, let me find if we look at maternal obesity, a very simple demonstration that maternal health affects and programs the offspring is this survival curve from Rebecca Reynolds in Edinburgh, Scotland. Here we can see the number of offspring surviving at each time of death. And here in green are the offspring of normal weight mothers. And here in black are the offspring of obese mothers. And you can see something like a 5-year difference at the end of life I'm sorry, I don't know how this, it seems to have a mind of its own, in the survival. And so right off the bat, we see that lifespan is shortened, is programmed by the maternal BMI. So one of the first sets of experiments I began to do was with Elena Zambrano, who I've worked with for 25 years in Mexico City on rats. And I'm not, unfortunately, going to tell you too much, if anything, about non-human primates here, because I think I can get over many more stories to you by talking about the rat work. And our obese model contains three groups. It contains mothers who are fed on the control diet before pregnancy, during pregnancy and lactation, and after pregnancy. And then we have a third group in which the mothers of, second group, mothers fed on a high-fat diet, the obese mothers. And then we have a third group, mothers who started off at their own weaning on a high-fat diet, the same diet that was used to make these obese mothers obese. But 90 days before we breed them, we put them on the control diet. It's a dietary intervention group to see if we can reverse or at least prevent some of the effects of developmental programming, extremely important in human clinical medicine. And I would call that the DINT group, the dietary intervention group. And here we can see that the important point of this experimental design is that all these three groups, this is extremely important, after weaning have the same diet. And so any differences in outcomes later in life have to be due to these differences in diet during embryo, fetal life, and weaning. And then we look at these animals mostly at 110 days of gestation. So let's first look at what happens to the offspring liver. The liver, of course, is an extremely important organ, performing both metabolic functions with lipids, carbohydrates, and amino acids, all forms of metabolite. And here we see the mothers who've been made fat. They have excessive weight gain, excessive lipids and glucose in their excessive lipids in their blood, in their tissues, and in their liver, and they become insulin resistant. They accumulate fat in the livers, and their offspring, the F1, accumulate fat and increase their de novo lipogenesis and reduce turnover of fatty acids. And these are some very basic measurements in the livers of the offspring. Most of the data now is going to be on offspring at 110 days of age. Our breed of rats lives about 850 days. So this is about a 20, 25-year-old human, not very old. So when you see some of these remarkable results, one will really begin to worry about programming by maternal obesity. And here we can see the liver weight in the offspring of the obese mothers is up. And when you intervene with the dietary intervention, you can reverse that. Similarly, the liver total fat is up in the offspring of the obese mothers and can be reversed. So we can see that there are lots of disadvantageous metabolic events happening, and they can be reversed by dietary intervention. So our overall hypothesis then is that dietary intervention, by transferring obese mothers back to their normal chow 1 month before pregnancy, would prevent adverse F1 male outcomes. And that's the hypothesis that we're going to test. What's happened there? Oh no, how is it? Okay, so here are more metabolic data. Here are the body weights, the adiposity index, the triglycerides, the glucose, the insulin, and the insulin resistance, all of which, except the actual glucose itself, because the body's compensating all the time by secreting more insulin, are increased and adversely affected in the obese mothers, offspring of the obese mothers. And we did RNA-seq on these animals, on their livers, and we found that there were a considerable number of genes which were differentially expressed. I'm going to try and keep to principles in this talk, and you may ask why virtually all the data is male. Well, the male always seems to take a bigger hit from developmental programming. It's as if the males are more stupid than the females. They don't try to compensate for the challenge. They just keep charging ahead in their growth patterns as they would if there was no challenge. Whereas the females are much more discreet about their and make many more compensatory movements, so to speak, in their metabolism. So they are generally affected, males are generally affected very much more than females. And here we can see when we compare the offspring of the obese mothers with the controls, 1,317 genes are upregulated, 48 downregulated. And that picture is greatly reversed by the dietary intervention compared with the MO. The major KEGG pathways that are changed by the maternal obesity in the offspring are, not surprisingly, pathways related to fat metabolism, biosynthesis of unsaturated fatty acids, fatty acid elongation, PPAR signaling, which is very important in terms of the turnover of fat, and fatty acid degradation. So these pathways are significantly affected and have several genes downregulated in the offspring of the obese mothers. If we look at specifically at some of these genes, we can see that a perilipin, this is mRNA and this is protein. Perilipin is increased in the offspring of the obese mothers in their livers, and that is reversed by dietary intervention. Perilipin inhibits, normally causes impairment of fat deposition. Here we have CPT1C, which is a major mitochondrial gene, and it is down as a result of maternal obese programming by maternal obesity. And it isn't really very much reversed by dietary intervention. That's the mRNA, and this is the protein. I believe now, after thinking about this for a very long time, all our cellular organelles are very important. But I know you do a lot of exercise at your university, and I cannot but believe that mitochondria are not one of the most important subcellular organelles. If you don't have a powerhouse to produce you energy, your cells are just not going to do well. And you'll see I'll introduce that concept a couple of times as we go along. So here are the offspring of the control mothers, a nice sylph-like rat. Here is an offspring of the obese mother, and here is an offspring of a mother who was started off on the high-fat regimen. Immediately she was weaned, but then was put back onto the normal diet. And that has restored this DINT offspring to something very similar to a control. One of the things about this developmental programming by obesity is that, unfortunately, we live in an era where we have one pandemic that ought to be enough for us, but we have an epidemic of obesity throughout the world, and something needs to be done about that. And now there are many interventions: resveratrol, DHA, exercise, which I will talk a bit about, and which, as I said, I know you guys have contributed greatly to, and probiotics. So in Dr. Zambrano's lab, there are a lot of studies going on these different exercise effects, because as with every drug, all drugs have side effects. There's no panacea, and people are very leery about resveratrol. It's very concentration dependent and can have very marked side effects. So that's programming by maternal obesity. What about the other end of the spectrum, maternal undernutrition? Well, in the fall of 1944, the Allies tried to seize a bridge over the River Rhine in Europe and bring the Second World War to a premature conclusion. There was unfortunately a lot of trouble with that airborne assault. I won't go into the details, but it failed. And the Dutch resistance had risen up to help the Allied forces. And as a result, the Nazis exacted retribution on the Dutch population. And the Dutch population in the west of Holland was essentially starved. All trains were stopped. No food was sent around. It was a brutally cold winter, and you need calories in the winter. And so there were many women who were pregnant at that time who had inadequate food supplies. And the Dutch keep very good records, have always done so, of pregnancies and details of life course health afterwards. And here is some pictures from some of the archives. The average diet was 400 calories a day. Here are some of the so-called daughters of the Dutch hunger winter. And here are the records from the Wilhelmina guest house, which is the bigger hospital in Amsterdam. So it was a very big occasion. And there are studies by the Dutch group which have extensively studied the children of the Dutch hunger winter, especially Rebecca Painter and Tessa Roseboom. Those children are now 65 years old. And so we know what sort of health they have experienced during life. And here is one example of the effects of the Dutch hunger winter on the offspring's health. And it's a very important slide, this one. I'm going to leave my slides. I don't mind you having them. I've put this little, it's not on here, this little cover of the book on all the slides, which are mine. And you're very welcome to use them quite freely for anything you want. The others, I think, may be questions of copyright, so I distinguish between the two of them. So here again is here's a sort of cumulative curve, but the other way around this time, not accumulation of death, but accumulation of the incidence of coronary artery disease. And you can see that the women who are really near the end of their pregnancy, so only had their last trimester, the last 3 months in the Dutch hunger winter, here is their incidence of their children's coronary artery disease. But here is the incidence of women who didn't become pregnant until the Dutch hunger winter was well underway. And so their fetuses were exposed early in pregnancy. And you can see that the incidence of coronary artery disease was very much greater. And this is the classical picture which everybody bears in mind when we look at the sensitivity to adverse outcomes from programming. Very much depends on the stage of development at which the offspring in the womb experiences the deficiencies. And that is something always to be remembered. And you might test yourselves as to why the babies who were nearly delivered when the Dutch hunger winter started had the best outcomes, where the babies who actually became pregnant during the hunger winter had the worst outcomes. Now, that is undernutrition. And here's another recent very interesting undernutrition study by Adam Watkins and Kevin Sinclair from Nottingham in England. I think I'm sorry, I apologize. I think I'm using this, Here we are. So what they did was they took normal, healthy, pregnant mice.
Peter, if you right click, you should be able to activate like a laser pointer. So if you right click, or like
I'm using my
yeah, but if you right click, you should be able to turn it into a laser pointer. So right click.
Can you see that?
Yeah, but it disappears after a few seconds of not moving it.
What I do when I right click is I send it on one.
Oh, never mind then. That's fine. Sorry.
I apologize. So Adam Watkins and Kevin Sinclair in Nottingham in England took normal mouse mothers and undernourished mouse fathers. And what we can see here is that here is the embryo developing, and this is the control father and the control mother. And you can see lots of blood vessels in the uterine lining. Can you actually see that pointer?
Yeah, when you're moving it, we can see it. Yeah.
Okay, so here is the here are the blood vessels in the uterine lining, and here are the cytokine and other molecules and chemokines that the mother normally secretes on the uterine lining. When the father is an undernourished father, then there's not so much angiogenesis, and there is not so much secretion of these cytokines and chemokines. And the offspring are just like the children of the Dutch hunger winter, are more susceptible to diabetes and other conditions. Now, this is a very interesting episode of my life, because when the first edition of the book came out, I went on the Diane Rehm show in Washington. And Diane Rehm, I think Diane Rehm and Fresh Air with Terry Gross are probably the two biggest talk shows on National Public Radio. And Diane Rehm always asks an expert onto the talk show to sort of balance the speaker. And that's a good idea, very good idea. So when I stopped, the expert on that occasion was the medical editor of the Washington Post. And she said to me, Dr. Nathanielsz, you can't tell this story. It will make women feel guilty. I replied that I have to tell the story for many reasons. First, the biology is so fascinating. Secondly, programming has enormous implications for the health of our children and grandchildren. And thirdly, if we understand programming and understand things like diet, the correct dietary interventions, we will be able to lessen the health costs, the healthcare burden throughout life. And that, of course, has great economic implications. Well, of course, that was 1999. And Kevin Sinclair and Adam Watkins' experiment that I've just taken you through was not available. So now we can say that this is a two-person job. And as I said to her, to the lady, I won't mention her name, it's not fair on her. I remember it very clearly. As I said to her, pregnancy is not a solely female responsibility. What are you talking about? The pregnancy is a responsibility of society, not just the woman. And now, of course, we've got biological evidence that the male effect of developmental programming. Actually, they've done a very interesting further experiment where they separated semen and sperm and did the 4 mixes, normally fed father's semen, protein restricted father's sperm, and then you've got 4 combinations. And what they've shown very elegantly is that many of these effects are non-genetic. They are caused by deficiencies in the semen itself. So for all of those of you PhD and less joining in on this discussion, there's an amazing area of work for you to do. I mean, if I could buy it, I'd switch places with you tomorrow, in fact, today. There's so much of excitement to happen in the near future. So programming tells us that we are nature and nurture. we are the product of combination of both genetics and epigenetics. I had a very good friend in Dallas, Gene Wilson. He was editor of the biggest textbook of endocrinology and other big textbooks, a real polymath. And I told him that the developmental programming will mean that the switch will come from nature to nurture. That there's just been, for too long, people have been able to say, there's a gene for this and a gene for that, and that gene looks after this altogether. I know very few conditions other than Huntington's chorea where you will go mad at the age of 40, regardless, unfortunately. We are a combination. We are a phenotype that is an epigenome, that is a combination of nature and nurture. And here's a good example from Rob Waterland in Houston in Texas. Folic acid is a B vitamin that plays a central role in regulating DNA methylation inside the cell. And that DNA methylation is an epigenetic change. These are all mice from the same litter. And the mother has been given extra folic acid. And usually these agouti mice are the color of the lightest ones on the left-hand side. But when you give a lot of folic acid, they generally become darker. One interesting thing which will enable you to investigate or sharpen your ideas about fetal development is these are all in the same uterus. The mother's presumably got the same amount of circulating folic acid in her blood. It's very interesting. Make a list of all the different factors that are going to affect why one mouse on the left-hand side there is light brown and one mouse on the right-hand side is dark brown, despite the similarity in the mother's blood levels of folic acid and the fact that they're in the same uterus. So I'm sure you've seen this before. Here is the DNA double helix, and here are the cytosine, guanine, adenine, and thymine nucleotides. And what you can see is that sometimes in some places, a cytosine and a guanine are next to each other, and they form what's called a CPG island. In fact, there are usually many more than that. And it's at this location that in those agouti mice, folic acid will help to put down methyl groups, methylate this portion of the DNA, and generally inhibit the transcription of that particular gene. So there's a lot more than just having a particular gene to how it's expressed. This is another example of epigenetic modification, and I think a very fascinating one. These pictures were kindly given to me by Richard Maleshka in Australia, who's a real expert on the honeybee. And here you can see the honeycomb with all the little cells, but one cell is much bigger than the other. And here is the larva that's going to become a queen in this big cell. And if the larva in one cell is fed royal jelly, that royal jelly stimulates the production of juvenile hormone, JH here, so that it rises above a certain threshold, and that bee becomes a queen bee, and the rest become worker bees. Here's the same thing demonstrated in a different way. Here we can see the larva, which has been selected to feed on royal jelly. Here we can see the royal jelly, which has a different composition. And here's the queen forming, and here are the workers. It's very interesting. I'm going to talk a little bit about developmental programming and aging. The queen bee lives 2 years. The worker bee sometimes doesn't reach 2 months. So here again, we have an example of nutrigenomics and developmental programming and interactions of programming and aging. I think it was the 1950s that a very famous biologist called Waddington had this concept of what he called his epigenetic landscape. And what he said was that we're like a ball here at the top of this landscape, and we run down the landscape being influenced by all the other factors in our environment, that if we bump here, we get diverted that way. This is diagrammatic, of course. If we bump here, we get diverted that way. And so gradually, the ball at the end becomes very different from the ball at the beginning. And that is very well elucidated in the in the honeybee story. So you are not a genome. You are an epigenome. And that's a very important thing to remember in terms of your functionality, all of us. I'd like to take you on another experiment of developmental programming, which I find extremely fascinating, from Dr. Dino Gisani at Cambridge, on the effects of fetal hypoxia as a programming event. And Dino was a postdoc of mine. He was born in Bolivia, so he knows this stuff, having been raised at 15,000 feet. And you can see here the eggs he works on. If you peel off the top of the egg, you can see the thin membrane stretched across the air sac. That air sac is full of air, and you could, if you want to, equate it to a sort of lung. And here, if you peel away this membrane, you can just see below it, and see even better here, the chorioallantois of the egg with all the blood vessels that are going to absorb the air. And that's the model he used. He also does a tremendous amount of work on sheep and rats. And what you can see here, I think, is extremely fascinating. Here is a newborn chick born of an egg from a mother who has lived at sea level all her life. And here is a newborn chick born of an egg of a mother who has lived at high altitude all her life. So hypoxia has certainly affected growth. Here is an egg of a mother who's lived at sea level all her life, incubated at high altitude. So they, of course, they're not exactly the same. They're not twins, but you can see that these are very similar eggs, and yet at high altitude, you get a much smaller chicken. Here at high altitude, you get a small chicken because it's at high altitude. And when you take that high altitude egg or a similar high altitude egg to sea level, you get a much bigger chicken. And in fact, if you can, the one interesting comparison is these two. Both of these are at sea level, were incubated and delivered at sea level, and yet the hen's egg that of the mother who'd learned to live at high altitude and conserve her oxygen and had lots of tricks for using oxygen is very much bigger than the chick from a mother whose background of her ancestors is all at sea level in Bolivia. Just because it's bigger doesn't mean to say it's better. It might have a worse cardiac system, might have worse cognitive function and what have you. But it is very fascinating. And there are human parallels here too. In La Paz, the capital of Bolivia, the offspring of mothers of Indian extraction who've lived up there for many, many centuries generally have larger babies than the offspring of European and American type mothers who've only just moved up there in the last 2 or 3 family generations. So hypoxia affects growth. And here we have an experiment by Steve Ford, who I worked with closely for 10 years, in which he did an embryo transfer experiment. And what he did was he investigated small Chinese pigs, Messian pigs, and large Yorkshire pigs. And when he took Messian embryos and put them in a Messian mother, small pigs here, and in the Yorkshire mother, the similar genetic material always grew much bigger in the Yorkshire mother. And similarly with the Yorkshire embryos, they always grew much better in the Yorkshire mother, showing a fundamental basic principle of fetal growth, that the mother constrains the growth of the offspring. And that, of course, is very wise. You don't want to grow a fetus larger than you can deliver. One of the last stories I'd like to tell you is to look at this interaction of aging and developmental programming. Here you can see, this time I am including some female data because I think it's very important to realize that there are sexual dimorphisms in virtually all developmental programming and developmental processes. For those of you wanting to go into a scientific career, just remember these words. In 95% of grant applications, unless you look at both sexes, you will not get funded. The NIH is now very strong on that we need information from both sexes. And here you can see, the liver fat at 110 days of life, 450, 650. Remember, they usually die about 850. And here you can see the growth, the aging-related increase in liver fat. But in the offspring of the obese mothers, this is the aging-related trajectory of rise of liver fat. And I mean, even by 110 days, it's going to be higher than it ever gets in the life in a normal male, in a male offspring of a normal mother. Here, triglycerides, the same. They programmed females also exceed non-programmed females, but not by nearly as much. And one thing I strongly recommend, that about the first thing you ever ask yourself when you look at a graph, you'll see how much more affected the males are if you look at the axes, which are not exactly the same.
Hey, Peter, I just wanted to give you a heads up. We have about 10 minutes left, and then we would have 5 minutes for questions. So I see you have
okay. Well done.
I
Yes, thank you. Thanks so much. Okay. Yep. Very quickly, I will draw it to conclusion. And this is nitrotyrosine, which is a marker of ROS action. And again, it increases in control mothers as their offspring age. It increases, but it increases much more in the mothers of obese mothers, the offspring of obese mothers. I'm going to miss these. So let's stop here essentially, because I've covered the main points that I wanted to. And here you can see this cartoon which explains why with the offspring of undernourished mothers and the offspring of overnourished mothers, we get similar outcomes such as aging, because during development, the placenta looks after nutrients, glucose, oxygen, and all of those, if they're short, give rise to extra glucocorticoids. So if placental function is impaired either by maternal undernutrition or maternal overnutrition, you get similar responses in the fetus. Okay. So those are some of the stories I tried to tell in the book. As I said at the beginning, that journey is the most important journey we will ever make. And that's why I find it so fascinating. You can look back at some of these. I'm sorry I haven't had time to go through them all, but as I said, I'll leave the slides for you. Okay, let's stop there and ask for some questions.
Yeah, that was awesome, Peter. I'm excited. I mean, I read the book. I never put reviews on Amazon, and I actually wrote a review, and it's cited as one of the better reviews from the United States of your book. So I was really excited to read the book. So thank you very much. There are multiple people on here. If you want to jump in with a question, please go ahead. I have lots of questions for Peter, but I would like to give everyone else the opportunity to ask them. And these can be broad-ranging questions about developmental programming as well. So if anyone has a question, please pop up or put it in the chat.
I have a question. CD Whale Rashid, I really enjoyed your talk. My question is about interventions during adulthood. I mean, you showed a lot about the effects of parental exposure, but are we doomed? You know, if my mom smoked when she was pregnant with me, which, you know, what she did, is there anything that I can do to help prevent, you know, some of the adverse consequences?
Super question. And in fact, there were some slides, and I sort of feel like a traveling salesman sometimes. There's quite a bit in the book about this in relation to exercise. We exercised some rats, pregnant rats, obese rats during pregnancy, and that ameliorated the outcomes. More interesting in many ways, I'm sure Kevin's very interested in this, we collected some offspring of obese mothers, and we exercised them at the equivalent of a human age of 40, and they were still improved. And I think we put in the title of that paper, "It's Never Too Late." And as you know, you probably got one of my hobby horses already from my talk. Until I started breaking legs right, left, and center, as I got older, I used to be an athlete. I couldn't let a day go by without running 6 or 8 miles. I didn't feel good. And I do believe that exercise is one of the best things for our lives. And I think it's a very good recuperator. You don't have to run 6 or 8. But yours is a superb question. How reversible are these negative effects? And of course, there's a lot of research going on into that. One of the slides I had later on was on resilience. I find that the term resilience extremely interesting. Why are some people more resilient to things than others? So thank you. That's a terrific question.
Does anyone else have a question? Well, CTWA, I'm sure you have another one as well. Go for it.
Well, yeah, I was just wondering, you know, people can do all types of manipulations with placenta, with early embryos. And because males are so susceptible, do we know if it's the placenta or if it's the inner cell mass? You probably can't switch the two and have like a female placenta with a male inner cell mass or anything. But do we know if it's really the placenta or if it's the baby itself?
That's another fascinating question. I can't answer that, but I can tell you one interesting experiment, which a guy called Tom Jansen from south of England did. He showed that when you undernourished pregnant rats for just 3 days, there was a change in the ratio of the inner cell mass and the placenta. So right off the bat, there are changes which are going to have differential effects. So it's a super question. There's one thing I think that does need to be said at this stage, and that is the human race wouldn't need to be around. I wouldn't be around if it hadn't been managed to deal with these things during hundreds of thousands of years. But unfortunately, we now want to live to 70, 80 or so. In the past days, you know, in old days, it's 40, 50, and that was it. So I think this is one of the greatest stories, developmental programming, from the societal point of view. And unfortunately, there's a lot of work still to be done. I tried to get my dean at Texas to look for some rich donors for us, and he said, "Oh, no." He said, they don't want to interest in obstetrics and pregnancy. They're interested in cardiovascular disease and cancer. So I do this. Perhaps I should never do it. Right behind the deans, right shoulder was a picture of these 3 very beautiful daughters with their children. And I said to him, I said, so we're not interested in them? Is that right? I got, not only did I get 0 brownie points, I got negative brownie points.
Yeah, well, yeah, I used to try to make the argument that that parents love their children more than anything, and then except for their pets, right? So I used to make that joke, and that one never goes over very well either. But so Peter, there's a question in the chat here from MJ Howard, and the basic question is does the system ever reset, right? So if the new normalcy becomes overweight and obesity, will we then evolve to then, I guess, be able to reset that system to where then there's not the negative effects from the maternal obesity, or how do you think that works? And
well, that actually is a question you could do a lot with. Thank you very much. I'm afraid my crystal ball isn't quite big enough to really answer that with confidence. But I think from what we know about the endothelium of arteries and the deposition of fat in arteries, they don't recover too well from that. I think that damage, I know the NIH is very interested now in regeneration. I think it has an Institute of Regeneration now. But I think that would be the wrong way to go. I mean, it's interesting scientifically, but I've always believed, fortunately, I had parents who always believed that prevention is better than cure.
yeah, absolutely. I mean, same with exercise in adulthood, right? Like it can prevent obesity, but it doesn't do a great job of removing any obesity that's already there, right? So prevention, but it's not the best intervention always. But so that was a great question. I have a question, I guess, about maybe a more short-term reset to normal. And a lot of the stuff that you showed is fascinating, especially with the eggs and the chicks and I guess they probably have catch-up growth, but that's another conversation for catch-up growth. But I guess I was surprised with the rapid response where within an egg, you could move it to high altitude or low altitude. And that does suggest it's a very rapid response. So what if a human mom moved from Denver to Atlantic City, New Jersey? Would those altitude changes, even within the course of her own pregnancy, be affected?
That again is a
Where's
fascinating...
your crystal ball, Peter? Where's your crystal ball?
Yeah, well, what you're really addressing, I think, I haven't thought of it in this way, is what's the hidden potential? What's the reserve?
Yeah.
Because that high altitude chick, which would normally have been destined to weigh, shall we say, 20 grams, now you take it down to sea level, it's going to weigh 30 grams. So it has 10 grams of reserve there. And understanding how to unleash that reserve is, I think or hidden potential, as I called it, would be very, very interesting. How one would do it, I just don't know.
Yeah.
And again, I think that's where there's now a society that looks at resilience. That's again where resilience, what enables you in the developmental programming field, you have a thing called the second hit, where you have a problem during development and then you go and smoke later or something, and you get a second hit, and how resilient you are to that second hit. And I am sorry, I don't usually go over time. I don't know what it was, but there is a slide in there if you go and search for it, for those who are interested, on this issue of resilience.
Yeah, well, I think I gave a long introduction and we took a minute to get started. So
no, you're just too kind. I don't allow you to take the responsibility.
I guess the other question that I have, if anybody has more, they can either put them in the chat or raise their hand, because I'd love for someone else to pipe in. But I guess for me, it's the time of exposure. I mean, you showed a nice example of the early high-fat diets or the early Dutch famine, which is still fascinating to me that anyone can survive on 400 calories a day for an extended period, because I've had well more than 400 calories and I can't wait for lunch. Anyway, I guess the question is like, or maybe it's not even a question as much as a comment, but like, we don't know that even for smoking, right? So if a parent quits smoking prior to pregnancy or quits smoking during pregnancy or starts using e-cigs or some kind of nicotine replacement therapy, a lot of that data is not out there. I mean, it seems like common sense, but it's not. There's not actually evidence-based research that's out there that's overwhelmingly convincing, you know? So I think, like you said, there's a ripe area for a lot of research if someone's coming into this field. I saw
Exactly.
Pat, did you want to actually ask a question? Because I did not ask a question.
Yeah, no, thanks. That was an outstanding presentation. I really appreciate it. I'm fascinated with either rescue or intervention methods here because it's very clear F1 has a problem during development. But if you intervene yourself, F1, is something still cemented in your genome, epigenome, that is passed on F2, F3, and so on? So are we doomed in that sense?
Yeah, that's terrific way to look at it. I was very interested at the beginning about smoking, and I think I told you one of the things which I've enjoyed about being asked to do this is that I'm going to have to go and talk. I mean, I had another slide, but I won't try to go for it. There's a classic slide of a pregnant woman showing that when she smokes, 3 people are affected. I'm sure you've seen it, that 3 people are affected. So not just the baby in the uterus, but the genes in the gametes that are going to be the next generation. So where's the damage there too? You know, how can that be ameliorated or prevented? And I think that I don't know of any experiments on that at all. But recuperation is, I think, a really big area for future research. But on the other hand, I don't know. I just wish governments in all countries in the world would get together and do something about prevention. We know enough to do prevention. In a couple of European countries, only now are they putting folic acid into the flour. They're just doing that after long discussions. But again, perhaps we better not go down that road after to wear a mask or not to wear a mask issue.
Well, Peter, I just want to say thank you. And I think you did this on purpose, frankly. Leave some slides, leave us wanting more, so that way we can invite you to Lexington, either in the spring or the fall, get you on campus, go visit some local establishments. We can take a tour. Alltech, that they have a much bigger touring facility now because they've got a brewery and also still making that scotch. So I think if you're willing, we'd love to get you on campus at some point and we can continue.
It would give some time to talk to people face to face, which is always the nicest thing. And that's really what I was going for. I wasn't being devious about this.
All right. Well, I think I can say for everyone, thank you. And we really appreciated it. And I can highly recommend his book. If you don't have the $8 or whatever it is on the Kindle, I will send you, actually, my wife will Venmo you the money because you're only allowed 1 Venmo account per bank account. But I will reimburse you because it's well worth the read. And I've just been texting a few people that I realized their wives are pregnant and they would be very interested in reading the book as well. And I will share.
Can I say something there, Kevin, very quickly, because it's very relevant. I've heard from so many people that they say, but I don't have a Kindle. And you don't have to have a Kindle. You can download the Kindle app from the App Store to your computer or your cell phone. It's wonderful. Now you can take 100 books around with you wherever you go in this world.
Hey, and one of my students bought your one from 1999, so I reimbursed her from that one and for the Kindle version. So some people just like to have it on paper, you know?
Yes, that's right. That's right. Well, thank you again. And
all right, let's catch up
you've
soon
all
in
got to
person.
get your lunch. And thank you so much for the questions. And Kevin knows how to get hold of me if you want to.
Yeah, anybody reach out if you want his contact information, or I really think we need to get you back on campus, Peter. It's 2011 was a long time ago. So let's get you back to Lexington.
Thank you very much. Take care, everyone. Stay safe. Stay safe. Bye-bye.